So, hello everyone. Welcome to our next part in the ECP 101 webinar series. Today focuses on cell and gene therapy in preparation for our two-day conference in March. For anyone considering a poster for the conference, the deadline is the 16th of February, and there is a separate ECP prize with cash and a glass trophy. So definitely put yourself in for that. We're very lucky to have Verena with us today, an expert in the cell and gene therapy field, bringing her experience of the area and some insights from Laverock Therapeutics. Please say any questions in the Q&A section and we'll ask them at the end. And without further ado, I'll hand over to Verena to give her very exciting talk on cell and gene therapy 101.
Thank you, Eve. Hello, everyone. So welcome to Cell and Gene Therapy 101. This webinar is designed for you, for early career professionals who want to understand one of the most transformative areas in modern medicine. So we talk about fundamentals, the potential, and the challenges of cell and gene therapy. Eve asked me to share a little bit about myself. So I'm Verena Brucklacher. I'm motivated and driven by the intersection of what I call the three S's. So science, strategy, and smart execution. So essentially bringing people and technology together to turn innovative ideas into real therapeutic products. And that really shapes how I make my decisions. So I started my journey with a master in neuroscience. Then I followed it with a PhD in neuroimmunology and a postdoc in immunology, during which I also became a mom of two. And after gaining deep scientific expertise during my PhD and postdoc, I became increasingly interested in understanding the impact this research could have. And that curiosity led me to join a biotech startup where I was exposed to the full drug discovery pipeline, which was actually quite interesting after academia, joining really a small startup to really see the whole process from start to beginning. And during my time there, I realized that strong strategic decisions rely not only on good science, but also on understanding the commercial and operational landscape. And to gain more experience in those areas, I moved to a life science tools and service provider. And while I was there, so in parallel to my job, I joined LVIC's volunteer community. And over time, I became more and more involved, and first through the science strategy team, where I was exposed to a wide range of scientific directions and built also a strong network, and later then as an LVIC board member. And the more I saw how strategy relies on operational and commercial decisions, both in industry and in a not-for-profit setting like LVIC, the more I wanted to strengthen that skill set. So I started an MBA to equip myself with the right tools to make really good strategic decisions and execute more effectively. And to put it all in practice, because theory is one thing, practice is another, I became LVIC's treasurer, guiding decisions from a financial perspective. And I re-entered the startup world by joining Laverock Therapeutics. And at Laverock, I've been able now to bring together science, operations, and commercial strategy work, which I really genuinely love. And you'll hear a bit more about Laverock later, as we are a cell and gene therapy startup. So yes, it's a busy time, wearing many hats, but it's absolutely possible. You can do it as well. And it's very rewarding. So unfortunately, there's only so much time we can cover in this webinar, time constraints, but fortunately, there is an LVIC cell and gene therapy meeting, Eve mentioned it, taking place beginning of March in Cambridge, which is a 2-day deep dive. And that's your chance to level up after this webinar from I get the basics to I meet the people rewriting health. Dave depicted here on the slide, and I have curated the meeting with superb support from our well-connected track chairs. You can see them here on this picture as well. And what can you expect from the meeting? You'll be surrounded by knowledgeable speakers. You'll experience insightful panel discussions, and you'll meet many interesting delegates and vendors. So if today's webinar sparks your curiosity, make the leap and join us in March. But now let's begin our journey into the science that is rewriting health, so cell and gene therapy. So imagine you're holding your newborn baby, you're happy, full of love and admiration. Then you hear the words no parent should ever hear. Your child has a rare genetic disease, one that will rob them of the ability to move, breathe, and live beyond their second birthday. So here in the photo, you see Sebastian. And what I described is exactly what his parents had to experience. He was diagnosed with spinal cord muscular atrophy, short SMA, which is a progressive muscular weakness and atrophy, leading to difficulty in moving, swallowing, and breathing. It is a devastating condition caused by a single faulty gene. So if Sebastian had been born in 2020, 6 years ago, Sebastian and his parents would have faced a heartbreaking countdown, knowing there was no cure, only palliative care. However, Sebastian was born in 2023, and there was hope because scientists had successfully developed a gene therapy designed to replace the missing gene. And it worked. So today, Sebastian is thriving, laughing, playing, and building a future that once would have seemed impossible. And that shows on one side how fast science is moving, and on the other side, how profoundly cell and gene therapy is rewriting the story of medicine. SMA was again in the public spotlight, I think recently, a couple of weeks ago. Even BBC News covered the story. And this again highlights just how rapidly the science is reshaping lives, and it also becomes more and more mainstream. So you will hear about Sebastian and his stories at ELRIC event in March, because we have invited his mom to talk about her experience. So Dharmisha will be there in March, and Sebastian's story shows what's possible when we tackle diseases at its root cause. So really going deep down and tackle it at a genetic level. But to understand why this is so revolutionary, we need to look at how medicine has traditionally worked. And then we can have a look how it is working now and how it can work in the future. So why treating symptoms isn't enough? So the old paradigm. For over a century, traditional medicine has focused on managing symptoms and not fixing the root cause. But treating symptoms is like mopping up water without fixing the leak. You're addressing the consequence, but not the actual cause. So even in the era of infectious disease, symptom management alone wasn't enough. The real breakthroughs came from root cause interventions like vaccines, antibiotics, and from behavioral changes like washing your hands. So fixing the leak really matters. And thanks to these measures, like diseases like polio, smallpox, and measles have been dramatically reduced. But they haven't disappeared. So COVID-19 reminded us that infectious threats still exist. So if fixing the root cause changed the game for infections, then imagine what it could do for chronic diseases today. So today, chronic diseases dominate, and they create lifelong dependencies on disease management. But science is shifting from managing symptoms to curing diseases. So cell and gene therapy targets the root cause at a genetic and cellular level, just as vaccines did for infections. That means the promise is one-time treatments that correct the defects. This is already real. So we will talk later about it. So CAR T for cancer, for instance, and I just mentioned the example of Sebastian, gene therapy for SMA, for instance. And looking ahead, so we're not only living in the present, but also looking ahead, these advances could move us beyond cure to prevention. So if we are going to rewrite the biology of diseases with cell and gene therapy, we need to understand the toolkit we are working with. So how do we do that? Meaning we really need to go back to the building blocks of life itself. And at the core of life are cells. So the functional units that keep us alive. Inside every cell is an instruction manual, the DNA, genes. And these genes tell cells what to do by producing proteins. So the workers that carry out every task from repairing tissue to fighting infections. And then there is the immune system, a system of different cells that form the body's security force. So cell and gene therapy works by going straight to these fundamentals, fixing the instructions, empowering the cells, and even reprogramming the immune system to fight back. So basically, scientists in cell and gene therapy explore the body's own toolkit, cells, genes, proteins, and the immune system. And when it works, well, then we stay healthy. But when it fails or misfires, disease takes hold. So in case it fails, nature doesn't give us a repair kit, but scientists have built one. And here's the fascinating part. Most of these tools aren't human interventions from scratch. They're inspired by nature, borrowed from microbes, and refined by chemistry and engineering. So let's look at some of at these tools a little bit more. So first up, editing, CRISPR. You might have heard of it. So to say, the molecular scissors. It was adapted from bacteria's defense system. Nature invented it to fight viruses. Humans repurposed it to correct faulty genes. Then there's gene addition. When a gene is missing or broken, we can supply a healthy copy using synthetic constructs, essentially giving the cell a fresh instruction manual. And of course, these instructions, they need to be delivered somehow. So we need a delivery system. And here scientists borrowed viral vectors from viruses, and chemists designed lipid nanoparticles, which we can use to deliver these gene editing tools or gene addition constructs to the cells. They are basically the couriers that get genetic payloads into cells. And finally, we also have cell engineering, and this is where we reprogram immune cells, turning them into precision weapons against, for example, cancer, but also more and more for autoimmune diseases. And you might have heard of, and I mentioned before, CAR T cells, where you re-engineer cells to be even more potent to carry out certain tasks. And these tools, like editing, adding genes, delivery tools, and engineering, are the foundation of cell and gene therapy. So now let's have a look a little bit more at gene therapy, because it's always called cell and gene therapy, but actually they are related, but they are also different. So let's first look at gene therapy. And gene therapy is all about fixing the blueprint of cells. So when a single faulty gene causes disease, the solution is simple in concept, but very powerful in impact. Replace or repair that gene. So let's take SMA, Sebastian example. It's caused by a mutation or a deletion in the SMN1 gene, which is essential for producing survival motor neuron protein. Without it, motor neurons die. So the impact is progressive muscular weakness and atrophy, leading to difficulty in moving, swallowing, and breathing. So what is the fix? The fix is gene addition, for example. So delivering a healthy copy using an AAV vector, a viral delivery system borrowed from nature. And the same principle applies to conditions like blindness and hemophilia. So one faulty gene and one corrected instruction delivered straight to the cells. And this is what's currently happening and what's already possible. But now imagine applying gene therapy to more common diseases. So that's where the challenge actually grows. So I have named here some of the challenges in terms of editing tools and delivery vehicles. So one of them is, for instance, efficiency. How can we efficiently get the genetic tools and the new instructions into cells? So currently the efficiency is still very low. Often it is single digit efficiency, or it is a little bit more like 10%, sometimes 20%. So it really depends. And then it depends also on the target organ, the target cell. And then specificity, how specific is the gene insertion as well. So if unwanted sites are edited, it's called off-target effects. And there are various different methods to detect these off-target effects. Eve, which you have seen at the beginning, and for example, works for a company called Broken Strings Biosciences that offers an approach where you can detect, for instance, these off-target effects. And I'm just highlighting it because there are lots of different technologies out there that actually enable cell and gene therapy as well. And one of them is sequencing. And another challenge is inserting the insertion size and the cargo capacity that also matters. And beyond the science, we face questions of safety, durability, immune reactions. Many of these tools we use are non-human, so we borrowed them from viruses and bacteria. So that means there's also an immune response involved and sometimes mounted against these tools. And then there are manufacturing challenges like complexity and also cost, because it's quite costly to produce these things and also to work at scale. But still, despite all of these hurdles, the promise is extraordinary. One-time treatments that rewrite the genetic code and can transform lives. And when you're interested in gene therapy, you can learn more when attending Alvik Cell and Gene Therapy event in March, because we have invited several speakers who will share the expertise. So some of them, they are here on the left-hand side of the slide listed. I'm really looking forward to hearing what they will share. And now we're moving on to cell therapy. So if gene therapy is about fixing the blueprint, then cell therapy is about upgrading the workforce. So instead of editing instructions inside the body, cell therapy takes cells out, reprograms them outside the body, and then sends them back again stronger than before. So cell therapy work has been successful with various autologous cell types and modification tools. I have listed some of them here in the table. And maybe just let's look at one practical example. For diseases like leukemia and lymphoma, autologous T cells, so meaning T cells from a patient, are given new powers through CAR T technology. So that means that patients' own T cells, which are autologous because they come from the patients, they are taken out of the body, then they are engineered to recognize cancer using the CAR technology. And the CAR stands for chimeric antigen receptor, which is a synthetic receptor we add to give these cells their cancer targeting ability. And then think of it as upgrading ordinary immune cells into elite cancer fighting soldiers. Then they get back into the body, they get reinfused again, and then they can hunt down and destroy tumor cells. And to give you a very current UK example, for instance, as well, the NHS treated its first patient with such a newly approved CAR T therapy at the start of this year, so 1 month ago, with and I think that the first infusion was delivered 2nd of January. So it's really happening. It's not sci-fi. And there are also various different in vivo gene therapy approaches for T cell editing. But for now, we're just staying in terms of CAR T cells. We use it as an example for cell therapy. But it's also possible to do it now in vivo. And there's a huge potential to expand into regenerative medicine as well. So not only oncology, also other diseases are possible to target with cell therapy. And here, for instance, an allogenic approach is very interesting. So this would be an off-the-shelf cell therapy approach where you take cells and you modify cells so that you could reinfuse them in various different donors, and you don't have to take first the patient's own cells. So the promise is huge, but so are the challenges. While delivery itself is easier than in gene therapy because you can take the cells out first, there are bigger challenges such as manufacturing, because you need to scale up, you need to maintain the cells in bioreactors, and also avoiding rejection is a challenge, and logistics, because you need to cryopreserve the cells and you need to transport the cells as well. And when you want to learn more about this topic, cell therapy, then we have also invited some speakers to our cell and gene therapy event. So some of the names are listed on the left side, and it also gives you the opportunity to speak to these presenters and ask them some targeted question, what you are actually interested in, and to learn more in general about cell therapy. So far, we have explored the big picture, the tools that let us edit genes, and I have shared some examples and what is possible in terms of cell and gene therapy. And these approaches, they are very powerful and they are also currently being used, but they are not perfect. So therefore, let's focus on gene editing for now to highlight some limitations and how they could also be overcome. So when we talk about gene editing tools and gene editing technology, then most gene editing technologies, they follow a one drug, one target approach. So for instance, CRISPR knockout, and siRNAs target one target at a time. But to silence multiple targets, multiple tools, each comes with its own delivery challenge, risk, or complexity. So either dosing challenges or multiple double strand breaks when you do several edits at the same time, and this could cause then unwanted side effects or apoptosis. But many diseases are not caused by a single target. So you need to target several several disease-causing genes because they are relevant. And our bodies are often master of redundancy. So if you knock out one gene, then another gene will take over. So therefore, therapeutic interventions have to target ideally pathways, so multiple targets at the same time to be really effective. And another limitation is that most technologies are non-context specific. So with that, I mean it's either an on or off. So you knock a gene out, then it's on, it's gone, and it's usually gone for forever. And gene editing technologies such as CRISPR knockout or prime editing or siRNA, they provide in addition no context-specific control. So they just apply the knockout or the knockdown regardless of the cell environment or any tumor microenvironment or activation status of the cell. They can't respond to signals like inflammation or tumor microenvironment or immune activation. So basically, when you only need a knockout or a knockdown of a particular gene. And another limitation is that most technologies are optimized either for ex vivo, so cell therapy, or in vivo, gene therapy approaches, and might be restricted to specific cell types, so only specific to neurons or only specific to T cells. However, scientists work on solutions for it. For example, the biotech startup Lavrock, so the biotech I've joined, we have set out to overcome these three limitations in a novel gene control approach. So Lavrock's tech enables us to overcome this one gene, one target approach. With one gene edit, we can target multiple genes, and a single edit can modulate several genes simultaneously. And Lavrock's tech enables also context-specific control, so meaning that multiple genes are not permanently silenced regardless of cell activation status or cell environment. Genes are silenced only when needed in activated cells or when sensing environmental cues. So this means we add an extra layer of specificity and of efficiency as well. And this really allows Lavrock to target multiple genes in the right cell at the right stage and to the right level at the right time. So really to have an intelligent system that can modulate genes. And this technology is suitable not only for cell therapy, but also gene therapy, and in addition is cell agnostic. So it works in various different cellular backgrounds. And I mean, I just use this as one example to show you that science constantly is tackling some of these challenges and is overcoming. And this is also what the speakers in the upcoming event will share. And so this technology is working, and we're currently generating preclinical data. And let's have a look what this technology could enable in practice. So we discussed CAR T cells before. Therefore, let's stay with this example and let's see when this technology is applied in a CAR T background, what impact it can have. So, as we have heard earlier, CAR T cells are currently revolutionizing treatments for certain blood cancers, and NHS has injected their first patients last month. But CAR T cell approaches, they face still some challenges. So, for instance, T cell exhaustion. So engineered cells, they lose their ability to fight over time because they are constantly activated and they are constantly overstimulated. And then cancer complexity is another challenge because tumors use multiple escape routes. So targeting one pathway or targeting one checkpoint target isn't enough. So therefore, Lavrock's technology addresses both. through an activation induced, so this is the context specific part, multiplex silencing approach. So not one gene, but multiple genes. And what that means is that we have activation-specific silencing. So Lavrock only silences checkpoint factors when T cells are active. So that means the intervention is temporary and reversible, so adapting to the immune system signals instead of acting blindly. And this prevents then exhaustion. And on the other side, Lavrock goes beyond a single target. It acknowledges the complexity of cancers, and it, at the same time, in an activation-dependent manner, will silence several checkpoint targets. So this maximizes then potency and also safety. And we are currently in the process of generating these data. So far, we have focused on the activation induced part and generating data for one target. And here you can see that on these graphs, we compared wild type CAR T cells with the activation induced silencing, which is depicted in dark blue. And in light blue, we have the CAR T cells that have a normal knockout of the target. And then we have the Lavrock technology, where we have an activation-dependent silencing of target one. And here we see that it reduces the tumor burden. Lavrock tech outperforms both unmodified CAR T cells and CRISPR knockout in reducing the tumor size. And why is that? Because Lavrock's T cells, CAR T cells, they don't get exhausted. So when you look at the graph at the very right, then this shows the expression of TIM3, which is an exhaustion marker. And Lavrock's technology shows fewer exhausted T cells compared to other approaches, meaning they stay active longer. So currently we're generating these data, or we have generated these data in heme tumors, and we are now also gearing up to show this is effective in solid tumors with various different targets multiplexed. Yeah, and I mean this is just a good example how smart tools can overcome several challenges in the future and provide even smarter, safer, and more effective cell therapies. So that was quite technical, and now let's switch gears again. So even if we have these tools, how do we make it real? How do we make it happen? So from an operational perspective. So therefore, we need to look at a cell and gene therapy biotech. So how does it work? And maybe when some of you work in academia, you might find this quite interesting. So how does it look actually on an industry level? So it starts with actually the disease indication, asking the right questions. When you have a certain technology, you need to find out in which disease indication could have my technology an impact. And is it feasible that my technology actually has an impact in a certain disease indication? And then you also need to assess the viability. Does it really have an impact for patients? And is someone willing to pay for it? And this then defines your actual disease indication. Is it oncology, or is it autoimmune diseases, or is it a rare disease? And then at the same time, for instance, if you target oncology, then there are various different sub-oncology indications which you have to investigate to see where your technology could have the biggest impact. And then next you zoom in even further and you decide what is actually your target you want to tackle. And then you need to maybe do a target identification, or you go for an already known target, and you can validate your target by both in vitro and in vivo assays to confirm that the biology really makes sense in line with your technology. And then comes at the heart of it is actually your technology, or your for cell and gene therapy, it could be a gene editing technology, or it could be a delivery system. And here you then apply this tool in various different in vitro and in vivo assays to show that your technology really has an impact. And then, of course, you do the validation, and you also do the preclinical testing to ensure safety and efficacy before moving anywhere near patients. So it's really a long way. And each step involves specialized teams. So automation and computational tools to really accelerate discovery and reduce errors. And it's a complex pipeline, but it's what transforms a concept into a therapy that can change lives. And every step you see here represents a unique career path. So cell and gene therapy isn't just one job. It's an ecosystem of roles: molecular biologists, bioinformaticians, process engineers, automation specialists, regulatory experts, and more. So at the bench and also off the bench. And as an ECP, you have options to specialize deeply in one area or become a bridge across disciplines. Whether your passion is hands-on lab work or computational modeling, manufacturing scale up, or clinical translation, there is a place for you in this pipeline. And the future of cell and gene therapy will be built by teams that combine science, technology, and also creativity, because there are still a lot of hurdles we have to overcome, and we have to build credibility as well. So that you get actually a better idea how life at the cell and gene therapy startup looks like. So these pictures, they are taken from the Lavrock Therapeutic Labs. And on the left, you see, for instance, an electroporator. So this is where we have cells in these cuvettes. And then we add the various different gene editing tools, so CRISPR proteins or guides, and then you put it in the machine and you apply a zap, so an electrical pulse, so that the gene editing tools go inside the cell. And on the right side, we have a sequencing device, because when you do cell and gene editing, then you need to do a lot of sequencing to make sure you edit the right sites, you don't have off-target effects, and what you did really has ended up in the right location and has the correct edit. And also computational work is involved, what you see in the left side, because you have to create your templates, you have to make your edits, and you have to do all the planning, the molecular planning on a computer as well. And also data analysis is very much computer driven. And on the right side, it's an example how you work in industry more at scale. So this is really medium low scale still. So you don't work in 6 well plates anymore. You really work in multi well plates. Here an example of 96 well plates, but it's not uncommon to work in 384 well plates as well. And because you have then many different plates, you can use various different tools to help you to do your tissue culture. So here, for instance, we have a semi-automated device which helps with dispensing medium. And these devices, they also exist in 384 well plate format, and usually you go through several of these plates per day. And then in industry, usually you also scale up, so you work even more at scale, and for that you use automation and robotics. So on the left side, also from our labs, an example of a robot which we use to help us prepare our sequencing libraries. I mentioned before, we do a lot of sequencing, because when you work in cell and gene editing, you you don't necessarily see under a microscope what has happened. So you really need to look at a genetic level, and for that you need to do sequencing. And we have usually many libraries to prepare, and this robot is helping us to do it efficiently. On the right side, an even bigger robot in a safety cabinet. So we use this device, for instance, to do splitting of cells. So it even splits adherent cells. It does medium changes as well, and it does imaging. So at the very right side of the photo, you see an incubator, and the incubator is connected with this cabinet with a conveyor belt. So it is actually really cool to see it in action. So basically the robot takes plates out of the incubator, they move along a little conveyor belt into the robot itself, and there the robot does the medium changes, does the splitting of the cells, and and then there is a little arm. I try to get the pointer working so I can show you maybe better. So here, there's a robotic arm as well. And this robotic arm is taking then the plates from here into this machine over there, which is an imager. So you can then also see the cells live. So you can check, are they clonally? Do they grow well? What is the density of the cells? And it's all happening in 96 well plates here, but the 384 well plates would be possible as well. And then the arm puts the plates back, it does its thing, splitting or medium changes, and then it moves back to the conveyor belt. So this is how it works. And there are also little cameras attached in the cabinet so that you can see from your desk in the office what the machine does, or also on your mobile device to make sure the robot works at night or over the weekend as expected. So that was just an example of how it works in the lab. And further, we have to move from the lab to the patient, and this means developing cell and gene therapies doesn't stop at the bench. We still have a long way to do after we have shown our technology has an impact, because then we talk about manufacturing, about clinical trials, and regulatory approval, and also patient access. And they all cost money. And here's what makes it hard and how we can actually overcome them. So this is discussed in this slide. In terms of manufacturing, you have high costs of raw material. You have the viral vectors, you have the reagents, the gene editing reagents are quite pricey as well. Then you need specialized GMP facilities, because these tools or these materials or these cells, they go straight back into a patient. Then it is sometimes it can be a fairly manual process still. I mean, there are more and more improvements, but some of the processes are still quite manual. Then you have sometimes a small patient population, which you have to overcome, especially when you work on rare diseases. That there's still a high uncertainty, because some of these technologies, they're pioneering new approaches. And then often you have a complex clinical trial design where you have to control for various different side effects. And then there are very stringent safety requirements, because this technology is still quite new. You have long timelines often to get your drug approved. And there's also global variability. So there's not a law or regulatory approvals which are international. So sometimes it really depends on the area and on different nations what types of regulatory approval guidelines they have. And then it's also about patient access, because it's the affordability challenge. These therapies, they're still quite expensive, and who is paying for them? The reimbursement hurdles. The governments usually pay for it, but then they cannot pay for all sorts of gene editing technology. So it's also, the industry is under pressure to make it more affordable. And then it's the logistics of personalized therapies, especially when you think about autologous therapies. You have to take the cells out first, and then you have to edit it, and no patient is the same. So you also have a lot of variability and patient to patient variability. But it's not all, it doesn't look too dire. It's not doom and gloom only. There are also proposed solutions. So for instance, we have automation and robotics, and they are getting better and better. Then there's the possibility of decentralized manufacturing hubs. So instead of producing cell therapies in a few large centers, you can think of a network of mini factories which allow to manufacture these cells. Then there are adaptive trial design. This means you can change the data as they come off from the patient, so you are more adaptive, you are more agile. And then there are real world evidence integration, especially when you work with ultra rare diseases, where you can, for instance, use health records from patients or natural history cohort data. So you don't have a completely controlled clinical trial like for other diseases. Then there's also the accelerated pathway possible for orphan drug diseases, so fast track, especially when there's a high unmet need. And then there's also harmonized global standards. So really different nations work together to harmonize these regulatory approvals to make it easier for developers of these drugs. And then there's also policy changes where governments and also systems, they try to shift to more to better frameworks which are more aligned with what cell and gene therapy needs. And then there are also innovative payment models. So one payment model is, for instance, that it's outcome-based reimbursement. Only when the drug is effective, certain payments are made. So there are various different options which are currently discussed. So just bear in mind that this technology, or this cell and gene therapy field, is still quite new. And if you want to learn more about it, then listen to our keynote speaker, Stephen Ward, who is joining us as well in March for cell and gene therapy in Cambridge. He's the Chief Technology Officer at the Cell and Gene Therapy Catapult, and he has more than 20 years of experience in cell and gene therapy. So now we move from science to society. So I think it's my last slide. And I think this is very important to discuss as well, because breakthroughs don't happen in a vacuum. They raise big questions about ethics, equity, and economics as well. So first, the affordability challenge. Why do cell and gene therapies cost over $1 million per patient? These treatments are complex, custom built, and resource intensive. Then we have the global disparities. So rich nations versus low resource settings. Who gets access when infrastructure gaps prevail? Specialized facilities, trained staff, cold chain logistics, so still exist. And here are also some ethical questions. Are we creating cures only for the privileged? Where do we draw the line between treating disease and enhancing traits like memory, strength, and immunity? And the ultimate question, if we can edit disease out of existence, should we do that? Can we and should we edit forever? So these are just some not only scientific challenges, but also sociential ones. I don't have a ready answer for that, but it's actually really important to think about it. And there are various different possible solutions like policies, pricing models, technology innovations. And it's just important to keep the conversation going and to think about it as well. And when you're interested about that topic, about also the ethical and social side of it, then we have organized a panel during the cell and gene therapy event in March, where we have invited different people who will discuss exactly this topic in a panel discussion. And here I'm at the end of the webinar, so I hope I could excite you for cell and gene therapy. Cell and gene therapy is already delivering cures for conditions one thought untreatable. And the next frontier is prevention, stopping disease before it starts. And I think for you, for ECPs, it's a brilliant opportunity to shape the future of medicine, whether you choose research or clinical development or manufacturing or policy. The world is your oyster. And thank you for joining today. I hope to see you all at ELRIC Cell and Gene Therapy in March, and we can continue the conversation there. So I've seen Eve popped up, so I'm happy to answer questions, be it related to career advice or cell and gene therapy science. Just ask away.
Thank you, Verena. That was a wonderful and really insightful talk. So starting with the questions, the first one is actually how can we submit posters, which I'll just quickly run you guys through. So the deadline for the ECP poster is the 16th of February. From that, you might get selected to do a flash poster presentation, which would be very exciting. The abstract is max 300 words and you need a title. So moving into the more topical questions, we have how do smaller companies that develop the therapies work with companies that might develop other aspects of the pipeline, such as manufacturing, delivery, safety? So yeah, it'd be really interesting to hear about how maybe potentially Lavrock Therapeutics kind of works with other aspects to develop your very exciting gene silencing technology.
That's actually a brilliant question, because especially for smaller startups, they work only with a certain amount of people. They only have a certain amount of budget as well, and they cannot do it all. So therefore, it's very important to work with reliable and good partners together. So it's all about collaboration. It's then reaching out to your existing network and also making new connections. Be really open-minded as well, because sometimes you might not know what actually could elevate your platform, what could elevate your technology. And also because there are constantly new technologies coming up. So then it's really important, for instance, if we have a perfect example, right? Because Lavrock is doing cell and gene therapy. We need a way to assess off-target effects. Eve happens to work for a company who actually does these off-target screens. So now it's exploring if the technology could add value to our platform as well. And it's the other way around for Eve as well, to see when we use actually the technology, because every company will use it in slightly different cells, so they will also learn at the same time. Or Lavrock is working with other partners. They have a particular technology, and then we onboard it or we license it out. And at the same time, also in terms of vendors, so where we get approached or we go to conferences, or we reach actively out when we see a need or a gap for a particular tool. So it's really a two-way route, I would say. And it's very, very important to dip into your network, to expand your network. And it's all about collaboration.
So what's been the most kind of exciting thing about your career in cell and gene therapy? What excites you about your job?
I think what excites me is really the prospect to cure diseases at the root cause, and maybe in the future to prevent even diseases. It is also, this is from a scientific level, but also from a more personal level. It is because cell and gene therapy is an area where you can combine various different disciplines. So I work with automation specialists, I work with bioinformaticians, with clinicians, so with really brilliant scientists as well, then you need also a good supply chain because you rely on various different tools as well. And they are quite pricey. So you need also a good supply chain that you don't overstock or understock, and you also get sometimes good deals. Then it's working with other people who enable these technologies as well. So again, it's actually collaboration. It's bringing it all together and working together as a big community to make it happen.
Lovely. Where else could Laverock's technology have impact? We saw a lovely piece about the CAR T therapies, but is there anywhere else that Laverock is expanding into?
So it is in terms of cell therapy, for instance, you could think about various other areas. So one aspect we, for instance, working on is iPSC-derived macrophages, where you can also have various different silencing um, uh, approaches which are cell state specific in terms of iPSC cells. They are pluripotent cells. You differentiate them, and then you need as a safety lock basically that they don't de-differentiate back, or they only silence when they are fully differentiated. And here, for instance, you could also have, instead of for the T cells, we had an activation induced approach. You could have a cell state specific approach that only these targets get silenced when these cells are really fully differentiated, and it locks them into a particular phenotype as well, because we know that when you are in the tumor microenvironment, then cell types, they switch their phenotype as well. So when you use, for instance, this context-specific technology, you can lock in T cells. And as I said, it's not only for cell therapy, the platform also lends itself for gene therapies. And currently, we're exploring various different indications. So this is exactly what I said, where you have to go through the process to see actually where your technology could have the biggest impact. This is then the bottom down approach, but you also have a top down approach. Where's currently the market? Where's currently interest? Where's the biggest unmet need? And then combining it both.
Thank you. And then we have kind of questions around would more extensive newborn screening allow for the development of cell and gene therapies to, be able to kind of detect and cure, maybe even at a pregnancy level? How do you work alongside these kind of understanding the disease that earlier on and maybe even preventing it?
Yeah, that's also a very good question, because often actually biomarkers are missing, because you need to be, when you work with cell and gene therapy, you really need to be very confident about what you're targeting. And ideally, you keep it simple. You have one particular gene, or even within this gene, it is very clear which base pairs you have to correct, or which part of the gene, or for instance, do you replace the gene entirely? So therefore, it is important to know the target really well, and then to make sure you can target it. Then in terms of, and for that, you need also screening tools, so sequencing early on, that you can actually assess if there is a gene deficit or not. And then also in terms of diagnostics, so currently for many of these diseases, there are not really diagnostic tools available yet, because then you need to have good assays and really reliable diagnostics as well to assess it, because you don't want to have any false positives as well. So this is currently all developing as well at the same speed, because yeah, you're absolutely, or the person who asked the question is absolutely right. It actually starts much earlier with diagnosing these diseases.
Well, thank you, Verena. Your talk has been super interesting. Such a lovely view of, the cell and gene therapy field and, how it interacts with all these different moving parts. So I'd just like to thank everyone for coming. It's part of our ECP Webinar 101 series. We'll have a few more that are quite similar. And just a final, everybody's welcome at the Cell and Gene Therapy Conference. Don't be afraid if you're an ECP, please come along. LRIG is also very kindly offering travel bursaries, the opportunity to apply for one if you put a poster. So that all the details of that are on site. So just helping with the cost if you are an ECP to get to our conference. We're really looking forward to it and hopefully really looking forward to seeing some of you there and of course your wonderful posters. So thank you once again, Verena, and thank you to all the LRIG staff that make this happen. All the best, and I hope you all have a lovely afternoon.
Thank you, Eve.